Hello interstellar everyone, welcome to class 12 biology. In our previous video we discussed about biotechnological applications in agriculture, in which we talked about production or creation of pest resistant plants such as pt cotton, right? And we also talked about the use of RNA interference in creation of nematode resistant tobacco plants. Now in this particular video we'll be talking about biotechnological applications in medicine, in which we will talk about genetically engineered insulin, gene therapy, and molecular diagnosis. Right. Now, when we talk about the application of biotechnology or recombinant DNA technology in the human healthcare, in the area of human healthcare, recombinant DNA technology has enabled mass production of safe and more effective therapeutic drugs. In other words, the drugs which are produced by use of recombinant DNA technology, they are safe and they are more effective compared to the conventional therapeutic drugs, chemical based drugs. Why? Because the recombinant therapeutics, they do not induce unwanted immunological responses, as is common in the case of similar products isolated from non-human sources. Now, in order to understand this second statement better, let us take the example of genetically engineered insulin. So insulin is a hormone which is involved in blood sugar regulation. A person suffering from diabetes, type 2 diabetes, the person needs to take regular shots of insulin hormone, because the cells, the pancreatic cells of the diabetic patient, they cannot produce insulin. So he or she cannot maintain his or her blood sugar level. So in order to maintain his or her blood sugar level, the patient has to take insulin shots. Now, where does that diabetic patient obtain this particular human insulin? He has to go and buy it from the market. And how does the insulin get into the market? It has to be produced by some kind of company, right? And in earlier days, these insulin which were given to the diabetic patients, they were extracted from slaughtered cattle and pigs, right? The cattle and pigs, they were slaughtered, their pancreas were removed, and from the pancreas they used to extract the animal insulin. Now that insulin is an animal insulin, not a human insulin. So therefore, when animal insulin is injected into a diabetic patient, a human diabetic patient, the human diabetic patient sometimes he or she shows allergic reaction. The immune system of that human being starts reacting in an undesirable manner. So it results in allergic reactions, right? So that is not desirable, right? So in order to avoid that, an American company called Eli Lilly in 1983, they prepared and introduced 2 DNA sequences into the plasmids of E. coli, thereby making them produce human insulin, right? Making them, I mean, these E. coli, which now has got plasmids with 2 human DNA sequences in it, that these E. coli, they start producing human insulin. And these human insulin can be milked from E. coli cultures in bioreactors, right? And now you may ask, why 2 DNA sequences? Why not just 1 DNA sequence and start producing human insulin? That is because in a normal human being, the pancreatic cells, they first produce insulin in the form of proinsulin form. It is not a functional protein yet, right? That protein has to undergo some kind of modification to become a mature functional human insulin, right? Inside the proinsulin, you can see 3 peptide chains: the blue one, the gray one, and the green one. The blue one is the A peptide chain, the gray one is the C peptide chain, and the green one is the B peptide chain. After it undergoes modification, it results in the formation of a mature human insulin, in which we only have the 2 peptide chains, A peptide chain and B peptide chains are there. They are linked together by disulfide bridge. The C peptide chain is removed. It is not required in the mature functional human insulin. Now, if the scientists, they want to produce a functional human insulin from the bacterial cultures, the scientists, they can get rid of this C chain. They can just get rid of this particular free C peptide. They don't need this one. So they took just the DNA sequence, 2 DNA sequences coding for A peptide and B peptide. They put it in the plasmids of E. coli. Thereby the E. coli will produce these 2 peptides, A peptide and B peptide, and these 2 will be joined together by disulfide bridges, right? And they can be used as human insulin, right? So that is the reason behind the introduction of 2 DNA sequences in the plasmids of E. coli to produce human insulin. I hope it is clear. Right. Now, this human insulin produced by genetically engineered E. coli, this genetically engineered insulin, right? They have got certain advantages over the insulin extracted from animals, right, such as pigs and cattle. And those advantages are you can produce large quantities of human insulin now, right? You can produce this human insulin in large quantities by use of bioreactors. We have studied about bioreactors before, right? And if you use these genetically engineered insulin, right, they do not elicit adverse immune response or allergy, right? There won't be any allergy because that is now a human insulin produced by the bacteria, right? It's a human insulin produced by the bacteria. So therefore, these genetically engineered insulin, they won't elicit adverse immune response. So you have to remember that these are the two advantages, right? If you use insulin extracted from pancreas of pigs and cattle, sometimes they induce adverse immune response or allergy. So the genetically engineered insulin, it was prepared so that they won't cause allergy. So I hope you are clear regarding genetically engineered insulin. So genetically engineered insulin is a form of recombinant therapeutics. Let us now discuss about gene therapy. So what is a gene therapy? Gene therapy is a form of corrective therapy for hereditary diseases in which normal genes are inserted into cells and tissues to treat the disease. So we have studied about hereditary disease in our genetics unit, right? So those diseases which are caused due to defects in the genes, they are called as hereditary disease. They can be passed down from parents to offsprings, right? So gene therapy is a sort of corrective therapy for those kinds of diseases in which normal genes can be inserted into cells and tissues to treat the patient. Right? One example is given in your textbook. One example of hereditary disease is given in your textbook, that is ADA deficiency. ADA stands for adenosine deaminase. Right? This particular enzyme is extremely crucial for the proper functioning of a person's immune system. So ADA deficiency, this particular disease will lead to immunodeficient condition, just like HIV/AIDS. So that is quite a risky situation. It's quite a lethal disease. So the first clinical gene therapy was done on a patient of ADA deficiency in 1990, and it was a 4-year-old girl named Ashanti De Silva. You don't have to remember her name. I just thought it would be useful for you to know that the 4-year-old girl name was Ashanti De Silva. Right. So the first clinical gene therapy was done on her. She was suffering from ADA deficiency due to deletion of a gene for adenosine deaminase. So in her treatment process, doctors, they introduced ADA cDNA into the lymphocytes of Ashanti herself, and after introducing ADA cDNA into the lymphocytes of the cDNA of lymphocyte Ashanti De Silva, the lymphocytes were injected back into the patient. Right? Thereby, these lymphocytes, they can produce adenosine deaminase, and she was treated. Now, there was a problem in this particular solution. It was a temporary solution because the lymphocytes, they have got a lifespan around 2 months, right? So the lymphocytes, they will die after 2 months. So she has to again go for the same process after 2 months, right? So that was a temporary solution. Now, it may be possible in future that if this particular disease is diagnosed quite early in early embryonic stages, a functional ADA gene can be introduced into the cells at early embryonic stage, and that could become a permanent cure. I hope it is clear, right? Now, this cDNA, it's a complementary DNA. cDNA stands for complementary DNA, and this cDNA, they are produced by reverse transcription process from the mRNA coding for adenosine deaminase, right? So such kind of DNA can be introduced into the lymphocytes of the patient, cultured in vitro, and these lymphocytes, they can be injected back into the patient so that the patient can be treated temporarily, right? So as soon as the lifespan of these lymphocytes, these transformed lymphocytes, they are over, the patient can revisit the doctor and the process can be repeated. Now, if you want to make the process permanent, right, if you want to make this treatment permanent, the disease has to be diagnosed at early embryonic stage, so that the cells at the early embryonic stage, they can be injected with the functional ADA DNA, right, functional DNA, and then the person can get rid of this particular hereditary disease, right? And the person's children won't suffer from ADA deficiency as well. So now there's one question for you over here. You can copy this down and try to find answers for that. This has been asked in previous board exams, right? How is gene therapy, the one which we have studied about just right now, it is better than bone marrow transplantation or enzyme replacement therapy, right? These are bone marrow transplantation and enzyme replacement therapy. These are two therapies which are used apart from gene therapy for the treatment of adenosine deaminase deficiency. Please, you have to find answers for this one. Okay. Let us now discuss about the applications of biotechnology and its tools in molecular diagnosis of diseases. Right. So in this particular process, the conventional methods such as serum and urine analysis, they are not good for early diagnosis of the diseases. Right. Serum and urine analysis, right, they are the conventional methods used for diagnosis of diseases. They are not good for early diagnosis of the diseases, right? So instead of using these conventional methods, we can use rDNA technology, recombinant DNA technology, polymerase chain reaction, and enzyme linked immunosorbent assay. They can help in early diagnosis. For example, we can use PCR or polymerase chain reaction for the early detection of the presence of pathogens within the human body, right? Normally what happens is the presence of these pathogens such as bacteria and viruses, they are suspected only when the person is showing some kind of disease symptoms, right? By that time, the viral load or the bacterial concentration within the person's body has become very, very large, right? So that the pathogens must have caused a significant amount of damage within the patient's body. Now, in order to avoid that, right, we have to detect the presence of the bacteria and virus in their lowest concentration. So that is possible by use of PCR. By use of PCR, we can amplify the genetic material of the bacteria or virus, thereby detecting their presence in their lowest concentration, so that we can avoid, right, certain kind of diseases early hand. So we can start treatment procedures. Right. So we can use PCR for that particular process. And regarding that, you can try to find out more information on the use of real-time RT-PCR in the diagnosis of the current COVID-19 virus, coronavirus. Right. You must have heard about two different processes, right? Rapid antigen testing and real-time RT-PCR. So you can try to find out some more information regarding RT-PCR in the diagnosis of the presence of coronavirus in people. Right, so that is one assignment. And this particular PCR method or polymerase chain reaction is also used to detect mutations in the genes, right? Mutations in the genes in suspected cancer patients. And it can also be used for the detection of other genetic disorders as well. So PCR is quite a useful tool in molecular diagnosis of different kinds of diseases, as well as the presence of pathogens within the human body. And then we have radioactive tagging method to detect gene mutations. Right. In this radioactive tagging method, a single-stranded DNA or RNA molecule is tagged with radioactive molecules, thus creating a probe. Right. And then this particular probe is allowed to hybridize to its complementary DNA sequence in a clone of cells. Now remember, these cells, they are being taken out from the suspected patient's body, and they are allowed to grow in vitro. Right. And the probe is allowed to hybridize to its complementary DNA in the clone of cells. And by using autoradiography, right, it's an imaging technique, we can detect the cells with the mutated genes. Right. Now remember, the cells with the mutated gene, they will not appear on the X-ray film because the cell containing mutated genes, they don't have the necessary complementary DNA sequence required by the probe to bind to. Right. So we can use radioactive tagging to detect gene mutations. Okay, so then next we have enzyme linked immunosorbent assay method or ELISA method. It is used to detect infection by pathogens. Right. In this particular method, the method itself is based upon the principle of antigen antibody interaction. Remember, antigen antibody interaction is very specific. Antibody is the Y-shaped molecule produced by B cells, right? So antigen is attacked by a particular antibody. A particular antigen will always be attacked by a particular antibody. So the antigen antibody interaction is quite specific. So enzyme linked immunosorbent assay is based upon this specificity of antigen antibody interaction, right? So infection of pathogen, right? If a person's body is infected by a particular pathogen, the pathogen can be detected by the presence of the antigens of the pathogen itself, or by the presence of antibodies produced by the patient's body against the pathogen. Right. For example, we have studied about the diagnosis of HIV infection is done by ELISA method. Right. So these are the tools which are used in molecular diagnosis of different kinds of disorders within the human body, right? For the presence of pathogens within the human body. And these tools, they are all present in the recombinant DNA technology. Okay, so in the next video, we will be discussing about transgenic animals and ethical issues associated with biotechnology. So please read about these two topics, and then you can watch the next video. Thank you.